today we're going to be focusing on pharmacogenomics and the considerations we should be making for Black patients and the Black community. So as part of our Black History Month celebrations this year, we wanted to focus on the positivity with the theme that is proud to be, and also have those broader discussions about advancing technologies in medicines and the considerations we should be making to ensure that it's inclusive and effective for everybody. So we've seen in other, like in medical devices, where actually it hasn't worked out so quite well with the pulse oximeter, for example. And also I know I've seen evidence of where things like hand dispensers don't recognize darker skin, and so they're not as effective. So it's thinking about that broader element of actually how do we make things more inclusive. And with pharmacogenomics, there's definitely an opportunity here now to make sure that we're thinking about it from the beginning. So we know that there are different genetic considerations that we need to think about when it comes to different racial groups. And we've seen that in practice. So for example, with ACE inhibitors, we don't use them in Black patients because there's a statistically significant rate of poor cardiovascular outcomes compared to white patients. So we need to make sure we're taking the same considerations into account with pharmacogenetics. So today I'm pleased to be joined by Raliat, who's a Chief Pharmacist and Clinical Director for Medicines Optimization at Barts Health, and also the Chief Pharmacist for North Thames Genomic Medicine Service Alliance, and also Innocent Asiimwe, who is a postdoctoral research associate at Liverpool University. So I'm really grateful for you two for spending your time this afternoon with me to talk about this. I'm just going to put out a disclaimer that pharmacogenetics is not, I can't even say it properly, pharmacogenomics is not my area of specialty. So I'll be learning alongside people listening today about the topic, and really keen to hear what we've got to say. But anyone that's listening, if you've got any additional questions, please do pop them in the chat box and we'll try to answer them. So to kick off, I just wanted to start with asking you if you could tell me a little bit about yourselves and the roles that you play in relation to pharmacogenomics. So Raliat, I'll start with you.
Okay, thanks, Aman. Yeah, as you said, my role is Chief Pharmacist for North Thames Genomic Medicine Service Alliance. North Thames, the alliance spans the whole of North London, Hertfordshire and parts of Essex. And the role of the alliance is to deliver for the people within our region streaming of genomics medicines. So as the Chief Pharmacist, I focus on and will be continue to focus on the introduction of pharmacogenomics testing, as well as the role of pharmacy within genomics medicine, and looking at how we might change our practice and what that means for pharmacy professionals as genomics medicines becomes moving. So I provide that strategic leadership and advice, and also working with a clinical lead pharmacist who will actually do a lot more of the implement of a lot of our agenda.
Brilliant, thank you, Raliat. And Innocent?
Yes, hello everyone. Innocent Asiimwe, a Ugandan pharmacist who has previously worked in Uganda and Botswana in both community and hospital pharmacy settings. I have currently transitioned in more research-based roles, and I am currently at the University of Liverpool working on an MRC-funded project. It's part of the Multi-Mobility Mechanism and Therapeutics Research Collaborative. The specific focus of the Liverpool team is on pharmacogenomics to identify genetic predisposing factors that lead to harm associated with drug-drug interactions in people on polypharmacy. Before this role, I completed my PhD with the University of Liverpool, and focus was warfarin anticoagulation in patients with cardiovascular disease in sub-Saharan Africa. Together with collaborators in Uganda and South Africa, we aim to develop clinical and pharmacogenetic dosing models to help improve anticoagulation control in these two countries. This project, which I'm still part of, is still ongoing. So recently, and together with Ugandan researchers at the Infectious Disease Institute, we have recently been exploring the genetic factors associated with TB-related clinical outcomes in HIV-infected Black African patients. So thank you.
Brilliant, thank you, Innocent. And the work that you're doing is like vital, because it obviously also expands to different countries as well, and using that research. So kind of just wanted to start a bit with a foundation of understanding of what is pharmacogenomics and why is it important to consider different races, different races and the genetic consideration. So Raliat, I'll come back to you.
So what is pharmacogenomics? Okay, so that's a question that I had to ask myself as well when I when I was looking into it. And I suppose in very simple terms, pharmacogenomics is the genetic reasons why a person does or how they respond to pharmacotherapy. So the genetic reasons underlying how a person responds to drug therapy. And those responses might be that are extra sensitive to a medicine, they are less sensitive to medicine, so they may not benefit as much if they're more or less prone to adverse reactions or to side effects. And that really is, I suppose, you say, no, what's the point of pharmacogenomics? It's understanding somebody's response to a medicine based on their genetic makeup.
Brilliant, thank you, Raliat. Innocent, would you like to add anything?
So to add on what Raliat has said, so pharmacogenetics comes from two words, pharma, which means drugs, and genetics, which relates to a person's genetic makeup. So it was coined in 1959, and later in 1997, there's another term, pharmacogenomics. So pharmacogenetics usually looks at one gene or a set of genes, while pharmacogenomics encompasses all the genes in the genome. So putting these small differences in definition aside, both terms are part of personalized medicine, which aims to improve the use of drugs from the present trial and error approach to a more precise one where drugs are given to patients who stand to benefit the most and experience the least toxicity.
Mm-hmm.
Yeah, in personalized medicine, we recognize that many factors which we can categorize as demographic, like an individual's age, clinical, like liver or renal impairment, and environmental exposures such as smoking, plus genetic factors will determine whether an individual will benefit from a drug or will suffer adverse effects. So if we consider differences in race, that is, if we look at the race-specific genetic factors that will influence drug response, we can further identify which patients will benefit from certain drugs and which individuals will not, which makes pharmacogenetics very important.
Brilliant, thank you. And then following on from that, is there actually any research that has focused on the difference in race, and what would be the result or consequences if we don't? I know, Innocent, you've done some research already and that you've already mentioned, but outside of that, is this something that's a common area that's researched, thinking about the difference in genetic makeup? Innocent, if you would like to start that.
So I've done a lot of research in warfarin. So let me first speak more about it, because it's actually regarded as the poster child for pharmacogenetics, and it's the most studied drug to date in the pharmacogenetics field. And it's an ideal candidate because it has dosing challenges like narrow therapeutic window, wide interpatient variability, and its mechanism of action involves more than 30 known genes, key among them being C9 and VCOR-RC1. So what research is there focusing on the different traces in warfarin? So as part of my PhD, we looked at part of the systematic review and literature research. We need to know what research is out there on warfarin and how it affects the different traces. And I could classify that most of the studies out there will focus on identifying the different genetic variants that are important in dosing. And after identifying them, you want to use those genetic variants to improve dosing or to help you identify which patients will benefit and which patients will not. So if I give an example of one of the systematic reviews we did, we wanted to find out how many warfarin dosing algorithms are out there in the literature, which have for the various populations. And by May 2020, we saw 433 warfarin dosing algorithms, of which 80% of these could be classified as pharmacogenetic algorithms because they incorporated genetic factors. However, there were racial disparities with only 27% of the algorithms being applicable to blacks. And if you, on the contrary, for instance, close to 50% of the algorithms were applicable to Asians and whites, so yet for blacks was only 27% of the algorithms. And for those algorithms, most of the studies included African Americans and Brazilians who are mainly of West African ancestry, and so it may not be generalizable to other African populations like those in sub-Saharan Africa, who come from a region where less than 1% of the algorithms were developed. So there is also a question is what happens when you ignore racial differences? And there is one key study which was conducted in the USA. It was called the Optimal Anticoagulation Through Genetics trial, the COAG trial. And in it, the investigators wanted to see whether if you give warfarin based on genotype guidance or clinical guidance, what option will be preferable to a patient. And it worked overall, it worked good for the whites, but for the African Americans, those were genotype guided, they performed worse than those who were clinically guided in terms of the time in therapeutic range. And the key reason was the study used the dosing algorithm, which had used data from white patients. So in whites, there are about 3 genetic factors which are important. But if you use only these genetic factors in black patients, then you are most likely to misdose them, which happens. Indeed, in a systematic review we recently published in Clinical Pharmacology and Therapeutics, we have shown that other genetic variants are important in patients of black African ancestry. And if you exclude these in dosing algorithms, you are likely to give these patients a wrong dose. So in addition to warfarin, there are other drugs which are dependent on pharmacogenetic guidance and include lipid-lowering agents like atorvastatin, simvastatin, antidepressants like citalopram, analgesics like codeine. There's clopidogrel, an antiplatelet, anticonvulsants like carbamazepine, and some estrogen-containing contraceptives. So if you use data from white populations to guide practice to black patients, you are most likely to give them wrong doses or wrong recommendations due to the differences in genes, in the male allele frequencies of the most important genetic variants.
Yeah, that's so interesting. Thank you, Innocent. And Raliat, did you have anything to add? Like, thinking about different conditions that black communities or African communities are likely to experience?
Yes, I suppose I'll probably answer sort of like on a wider perspective before narrowing down. So, I mean, you asked whether or not there's been research, and I think one of the key things that I would say is that yes, there is lots of research, because this is about how people's genetic makeup affects their response to drugs, and we know that people's genetic makeup is obviously linked to their race. Then, whenever research is done on pharmacogenomics, then it really needs to include whether which races were included in that research, because otherwise we won't know whether or not it is applicable to all to specific populations. So the answer is that yes, there is work done. However, and I think this is sort of like what Innocent said as well, there hasn't been enough work done to identify whether a genetic variation, for example, whether or not it means the same in different races, or in fact, if a specific race has got a different variant, which will affect their response to a drug. And I think that's where the gap in research is. It is known that that's a gap in research, but at the moment hasn't been enough work done on that. But you can't split the two things out. You can't do research on people's genetic reactions to a drug if you don't take into account what race that they're from. If we think about sort of like specific conditions now where Black people are more prone to. That now brings in the question of whether or not the Black community is participating sufficiently in research so that the response to particular drugs is actually identified. And I think Innocent mentioned clopidogrel, for example. So there's a very well-known genetic mutation, which means that some people are just not going to respond to clopidogrel. Black people are very, very prone to cardiovascular disease. So we need to make sure that whenever these genetic variations are identified, especially for those conditions, for example, cardiovascular disease, stroke, diabetes, etc., actually, are they also applicable to Black people? And the only way we'll know that is actually by having more people from the Black community participating in research, so that any genetic, because otherwise, all you know is that it's an issue for a specific type of community, people from a specific racial background, but not necessarily for all other racial backgrounds.
Yeah, no, it's really, and also then I suppose in practice, is making sure that then translate into different options. If you're thinking about a formulary and then making sure that there's different therapeutic options available, that will then be suitable rather than having one blanket option for everybody.
Exactly, and I foresee, it probably won't be in the near future, but I foresee that the same way as you said when you started, and then you said that we know that ACE inhibitors, different consideration in Black people. I foresee that with once you put pharmacogenomics into the mainstream, what you would be saying is that if somebody's got this specific genetic variation, then this is the dosing model for them, or this is the treatment. This is the treatment pathway for them. But actually what you need to know is to make sure that those genetic variations have been identified for all the different racial groups.
Yeah, no, that's a good point. Thank you. Innocent, I know you've already mentioned a bit about your work that you've done, but actually just thinking more broadly about it then, what did your results show you, and how can we apply that to pharmacogenomics more broadly, and perhaps for patients of an African heritage in the UK, and that we need to be considering?
So let me talk more just briefly about the WOPAF project. So we are developing and validating clinical and pharmacogenetic dosing models to help improve anticoagulation control in patients from Uganda and South Africa. So in the UK, for instance, there are very many dosing models for warfarin for white patients, but none for Blacks. And the same applies to Sub-Saharan Africa. So first, we did a clinical audit to see the quality of outpatient anticoagulation services in Kampala and Cape Town, and we found it was low with a median of 41% time in therapeutic range. So NICE, that is National Institute for Health and Care Excellence, defines optimal quality as 65% or more. So in the regions, the median is about 41%, which is quite low. So we want to try to find ways of improving the time in therapeutic range, and one way is to develop and validate models. So we've developed and validated the clinical model, which has been published, and we are currently testing it in a prospective cohort. And we plan to develop a pharmacogenetic model, which will incorporate genetic factors identified in the systematic review I mentioned above. So we are also conducting a genome-wide estimation study to further identify genetic factors that may be unique to Black populations. And lastly, we intend to develop a polygenic dosing model in the near future, as it's possible that response to warfarin treatment is influenced by a large number of genetic variants rather than looking at one or two variants with large sizes. In terms of applicability to Black patients in the UK, definitely an algorithm or model developed using data from Black populations is more likely to be applicable to Black patients compared to an algorithm developed using data from white populations. And we know that some UK patients who identify as Black may be admixed, that is of mixed ancestry, one parent Black, the other white or Asian. And to cater for these populations, we're also recruiting patients of mixed ancestry in South Africa. And lastly, and like for all algorithms, it is advisable to first validate an algorithm developed elsewhere before it can be recommended for clinical use in any given population. So any models from sub-Saharan Africa or from any Black population, if you are to apply it to any other population, you need to first validate it in your population before you can really recommend it for clinical use. Thank you.
Brilliant. Thank you, Innocent. It just sounds so obvious when you say it, doesn't it? But it's, you know, it's not something that we are very good at doing. And then I suppose, Raliat, bringing it back into that real life patient care, you know, how do you see this being translated into everyday care and, you know, that care being delivered?
So that's the, I suppose that's the $1 million question really for the genomics medicine service. So I would say that the role of the genomics medicines England and the service alliances is really to take the work that Innocent is doing and actually work out actually what does that mean for the mainstream and how do we move that into mainstream care. So the way that it works, the way that it's planned to work in the NHS anyway, is that when there's enough evidence that a certain certain pharmacogenomics test actually has real life applications to the way that you would use that specific drug, then it will then go into being commissioned. So that pathway would then be commissioned into actual real life treatment algorithms. So a good example, and in fact, you know, the main example really is the use of DPYD testing for specific chemotherapy drugs. So it's been identified that actually, if people do either have reduced amounts of a particular gene or they have none of that gene, they are much more likely to get severe toxicity. So what's happened now is that the use of that test before a patient is treated is now part of that treatment pathway. So it's sort of similar to, you know, that if you want to start this particular drug, you need to do their creatinine, you need to do their potassium. So it comes into the mainstream that way. So now, you know, so if as a pharmacist, you are trying to review a prescription or in fact prescribing for a patient who you know you need to give that, first thing you have to do is either order the test or check if it's been ordered. And then, and without the results of that information, you cannot appropriately prescribe for that for the patient in front of you. And that's how it. In some cases, there will be there may well be the way of actually screening for a panel of pharmacogenomics tests almost at birth, and that really is, and that will be with your whole, sort of like part of whole genome screening. So rather than being I suppose waiting until you need to prescribe the drug, what you might do, or what we might see then, is we're saying that there's all these common drugs. So at some point in somebody's life, perhaps at birth, you're going to screen for all of these drugs, for all of these genetic variants, so that if they do need to use these drugs later on in life, those results will already be there.
Wow, God, it's so interesting, isn't it? It like the future of medicine and how you can be one step ahead rather than reactive and thinking about what's coming.
Exactly. Yeah, and it's actually here now.
Yeah, and then thinking about, like, in terms of, I know we've been talking about inclusive professional practice in the profession and health inequalities, and how does that bring in like patients and their carers and all of that within that conversation?
Okay, so and I'm sure, you know, especially with Innocent's work in Uganda, etc., there's a large consideration around genomic literacy, and not only within us as professionals, but also how do we have that discussion and explain to our patients what it means to have that genomic screening test, bearing in mind that it has implications not only for the here and now, but actually for the rest of their lives, and also for relatives and family, because obviously it's about your genetic makeup. And one of the things that we do need to address, I think, quite rapidly as pharmacy professionals is where's the pharmacogenomic counselling, and how is that tailored again for different communities, and how do we explain what this means for different communities? So there's a whole we talk about health literacy, we're also talking about genomic literacy, but genomic literacy obviously needs to be tailored to people's cultural values, their beliefs, what this means for their families, not only just what it means for them right now. So there's a whole other area that needs to be discussed, because it needs to be very culturally sensitive.
Yeah. Innocent, did you have anything to add on that?
Not really. I totally agree with what she said. You have to look at the different cultures, at the different contexts, because some cultures they are afraid. I would say afraid of genetic testing. So if you ask for a blood sample, you want to test the genes. Some people might not understand what you're trying to achieve. So you need to look at the cultural context, and otherwise there will be difficulties in implementing the word.
Yeah, no, I agree. That's brilliant. I think this conversation's been great, and we could carry on all afternoon. I mean, my mind is actually blown. I'm not going to lie. Like, this is so interesting. But I think we should wrap it up, because I think that there's so many more things to consider. But this is just a beginning of that, isn't it? And all the things that we need to be thinking about. So I just want to say a huge thank you to both of you for sharing your expertise and the work that you're doing, and let's hope that we continue and making sure that the research is there and it's being done, and so we can make we're being inclusive from the beginning. So thank you to everybody that's listening, and I just want to say that we're continuing to there's a question that's just popped up, and I don't know if either of you wanted to answer it. It says, how will genomic screening data from patients be protected from commercial interests? I mean, that is such a broad question. Raliat, I'll start with you.
Yes, so I'll talk about this from the UK context. I am aware that we do have an international audience, but data security, ethics, confidentiality is a really important work stream from the NHS Genomics Medicines Unit. And I think that from one aspect, it really is no different from how anybody's health information is protected. Because we do have very strict protocols on sharing people's healthcare data anyway. What I would add into though in terms of genomic screening data is not just the commercial interest, but the fact that actually genomic data is also of quite a large research interest, because we're still an active area of research. We're still finding out things all the time. And so there's the translation of information that you pick up from just routine care for genomics. That's very helpful to research, and making sure that it's appropriately anonymized and treated according to all normal ethical protocols. And then how is that then moved into how is that of use to commercial interest? So I would say that it really is by using some very, very strict protocols and ensuring that confidentiality and ethics underpins all the work that's being done. And my personal view is that there needs to be a discussion about to whom does that genomic data belong?
Yeah,
that's the key question, I think.
Yeah, thanks, Raliat. Innocent, did you want to add anything?
So like Raliat said, as for clinical data, genetic data has their protective mechanisms in place. For instance, for the work we are doing, they're currently classified as clinical trials, and before you can go collect any clinical or genetic data, you have to go through several approvals, ethical approvals, regulatory approvals. It's not easy. It can take you several months before the regulators or ethical bodies are confident that you're going to use the data you're collecting for the right purposes. And there are mechanisms to make sure that patient data is protected and not misused.
Yeah, no, thanks, Innocent. And then the second question that's popped up is, how will patients right not to know be addressed? So people might not want to know their genetic makeup.
I don't have an answer to that, because I think it's at the moment an unanswered question. It sort of goes back to the whole thing about counselling and genomic literacy, and how you have those those conversations. And again, I draw parallels with people not wanting to know their diagnosis, for example, and how is that addressed. But I recognise that it's different, because if somebody finds, if you do have a genomic test, first of all, you have to consent to have that test, actually, and as part of the consent process, then people might want to know, or they can decide whether or not they do or don't want to know. But there is, again, that added thing of once that test has been done and that information is there, it's there for life, because it's unlikely to change. So there are parallels we can draw with what we do now, but then there are some added bits we need to consider because of those unique characteristics of genomic medicine that really needs to be object of debate. A large part of this does come down to, in fact, all of it, I would say, comes down to actually doing things in the best interest of the patient and making sure it's your patient's decision as to what happens, rather than making decisions on behalf of the person in front of you.
Yeah, brilliant. Thanks, Raliat. Innocent, anything to add?
No, thank you.
No, brilliant. Thank you so much for that discussion and the questions that have come in. It's been really interesting. So just to say that we're still promoting profiles of Black pharmacists and technicians and pharmacy staff across our social media platforms, which have been great to see. So if you want to get involved, it's not too late. Please do message us. We'll be having our co-hosted evening tonight with pharmacy technicians of colour, where we've got the amazing Dr. Joan Myers, who will be sharing her experience and talking about why she's proud to be Black. And then also we've got our next week, we've got a live event. It will be at 5:30, and we'll be looking at the importance of coaching and mentoring, and we'll be joined by Lely and Diane, and along with some other speakers. So please do join us for that. So just want to say again, a final thank you to Raliat and Innocent for today's conversation, and I will see you all soon.
Okay, thank you.
Thank you.
Bye.